Welcome to the Deep Dive, the show that gives you the shortcut to being well informed. Today, we're tackling a really fundamental topic: how life diversifies. We'll be looking closely at genetic variation and, crucially, the role of gene regulation in evolution.
That's right. Our mission today is really getting to grips with what's arguably the central challenge in biology,
Hmm,
understanding that link between genotype and phenotype. It's not a simple one-to-one map.
not at all.
And the core idea we're exploring is that, when new forms arise, when major changes happen, the key evolutionary action often isn't in creating brand new genes, but in changing how existing genes are controlled, the regulatory switches.
Okay, so let's unpack that. Where does the variation even come from? We're talking everything from small mutations, right, up to those master switches that kind of filled the animal kingdom.
Exactly. So the raw material, well, you've got the usual suspects.
Hmm?
Differences in alleles people already have, totally new mutations popping up, big duplication events.
Like whole genomes getting copied.
Yeah, or just chromosomes, or even parts of genes. And gene transfer, especially in microbes. But maybe most importantly for multicellular life, you have changes in how gene function is regulated at the DNA level, RNA, protein. All of it matters.
Let's start small then. What about the simplest change, just swapping one DNA base for another, a point mutation?
Right, the point mutation.
Hmm.
Seems tiny, but the consequences can be massive, or sometimes surprisingly beneficial. The classic example, of course, is sickle cell anemia in humans.
Ah, yeah, the textbook case. A single base change in the beta hemoglobin gene. That's it.
That's it. And that tiny change swaps out one amino acid, glutamic acid, gets replaced by valine, just at position 6 in the chain.
And that single amino acid difference causes the red blood cells to distort into that sickle shape, especially when oxygen is low.
Precisely. Which then leads to a whole cascade of health problems: blocked blood vessels, pain, organ damage. It's a perfect illustration of pleiotropy.
Pleiotropy meaning one gene influencing many different, often seemingly unrelated characteristics.
Exactly. But here's the evolutionary twist. You'd think selection would just eliminate such a harmful mutation.
But it hasn't, especially not in places with high malaria rates.
And that's the key: heterozygote advantage. People who carry just one copy of the sickle cell allele, the heterozygotes, they have significant protection against malaria.
How does that work?
Well, the malaria parasite infects red blood cells. But in carriers, those cells are a bit more fragile, especially under the stress of infection. They tend to rupture more easily, disrupting the parasite's life cycle.
Ah, so the parasite can't thrive as well.
Right. So in malaria-prone regions, the survival advantage against malaria outweighs the disadvantage of potentially having children with the full sickle cell disease. Selection maintains the allele at a relatively high frequency.
Okay, so that's pleiotropy one gene, many effects. It's good to contrast that with polygenic genes, right? Where it's the other way around, many genes contributing small effects to one single trait, like height or skin color.
Absolutely. And it's also important to remember that heterozygote advantage isn't the only reason a potentially harmful allele might become common in a population.
What else is there?
Sometimes it's just down to history and chance. Think about certain isolated populations. You might find unusually high rates of things like achromatopsia, total color blindness on some Pacific islands, or Ellis-Van Creveld syndrome, a type of dwarfism among the old order Amish.
So that's not selection.
Probably not adaptive selection, no. It's more likely due to the founder effect or a bottleneck.
Oh,
A small group of founders happened to carry the allele, or the population went through a sharp reduction, and purely by chance, that allele became much more common as the population grew again.
okay, got it. But going back to the idea of how mutations cause effects, it's not always about changing the protein's structure, is it? Sometimes it's about the amount produced.
Exactly. That's a crucial point, and it leads us straight into regulation. Take the IGF-2 gene in pigs, for instance. There's a mutation that doesn't actually alter the insulin-like growth factor 2 protein itself.
So what does it do?
It messes with the control element. The result is about 3 times more IGF-2 protein being produced in the muscle tissue. And what you get are these incredibly muscular pigs. The phenotype change comes purely from changing the regulation, the quantity.
That's fascinating. And it sets the stage perfectly for talking about gene regulation as this major evolutionary force.
Right. So regulatory mutation is basically any change that affects these genetic pathways or networks, the systems controlling when, where, and how much a gene is expressed.
Mm.
These are incredibly important for evolution, especially in complex multicellular organisms where timing and location are everything.
Where did we first figure this out? Wasn't it in bacteria?
Yes, the foundational work was Jacob and Monod's discovery with the E. coli lactoperon in the 1960s. Just beautiful, elegant work.
The lactose switch, right?
Exactly. They showed that if there's no lactose around, a repressor protein sits on the DNA and physically blocks the machinery needed to make lactose-digesting enzymes.
Blocks transcription.
Right. But if lactose is present, it gets converted into a related molecule, allolactose, which acts as an inducer. It binds to the repressor protein, changes its shape.
So it falls off the DNA.
Precisely. The block is removed, and the genes are switched on. It's a straightforward response to the environment.
Simple and effective for bacteria. But eukaryotes, like us, things get way more complicated.
Oh, massively more complicated. It's not just a simple on-off switch. Think of it more like an elaborate control panel with dimmer switches, timers, multiple inputs. There's regulation at many, many levels. It forms a complex regulatory code.
What are the main mechanisms?
Well, you've got control elements located right near the gene they regulate. That's cis-regulation. Then you have proteins, like transcription factors, that are made elsewhere but travel to control specific genes. That's transregulation.
Okay.
And then there's a whole other layer, RNAi regulation, where small RNA molecules themselves control gene expression, often by interfering with the messenger RNA.
And even after a gene is transcribed into RNA, there's still more control.
Absolutely. Post-transcription modification. Things like alternative splicing, where one gene's RNA transcript can be cut and pasted in different ways to produce multiple different proteins. Or RNA editing, changing the RNA sequence itself.
So one gene can actually code for several different products depending on how it's processed and regulated.
Exactly. And the discovery of small RNAs, like microRNAs, miRNAs, really highlighted the subtlety. These tiny RNAs don't code for proteins themselves, but they fine-tune the translation of messenger RNAs into proteins. They seem to be really important in the evolution and diversification of complex animals, like vertebrates.
Wow. So if you pull all this together, what's the big evolutionary take-home message?
It's that organisms, even very different ones, often share a huge proportion of their basic structural genes. The genes for enzymes, structural proteins, etc. The incredible diversity we see isn't primarily because they have wildly different sets of genes.
It's more about how they use those shared genes.
Precisely. It's about which genes are turned on or off, when, where, and how strongly. It's the regulation of that shared ancient toolkit that drives a lot of the diversification in form and function.
Okay, let's scale up then. From molecular switches to the grand plan, the body itself. Most animals are built along fundamental axes, right?
Yes, three main ones define the basic body layout. Anterior-posterior, head to tail, dorsal-ventral, back to belly, and left-right.
And when the genes controlling that fundamental plan go wrong, you get really dramatic effects.
You certainly do. These are called homeotic mutations, or homeosis.
Yeah.
It's where one body part gets transformed into the likeness of another. A part basically develops in the wrong place.
The classic examples are from fruit flies, Drosophila, like the Antennapedia mutant.
Right, where the fly grows a pair of legs on its head where its antenna should be.
That's bizarre.
It is. Or the bithorax mutations, where an abdominal segment gets transformed into another thoracic segment, resulting in a fly with an extra set of wings.
So how does that happen? What controls these big decisions?
It starts very early in development, even in the egg. Maternal gene products, called morphogens, set up concentration gradients, like the bicoid protein, establishing the anterior-posterior axis, and gherkin helping define the dorsal-ventral axis. These gradients tell cells where they are.
And then those cells activate specific master regulator genes.
Exactly, which led to the discovery of a key piece of DNA shared by these master genes, the homeobox. It's a conserved sequence of about 180 base pairs.
Found in lots of different animals.
Incredibly widely conserved, yes.
Okay.
This homeobox sequence codes for a protein segment of 60 amino acids called the homeodomain. And this homeodomain allows the protein to bind to DNA.
So these are transcription factors. They regulate other genes.
Precisely. These are the Hox genes. And what's truly remarkable is not just that flies, mice, humans, fish, we all have related Hox genes, but how they're organized and expressed. This leads to the concept of collinearity.
Collinearity. What does that mean?
It means that the physical order of the Hox genes along the chromosome directly parallels the order in which they are expressed along the anterior-posterior axis of the developing embryo.
Wait, so the gene for the headmost structures is at one end of the gene cluster on the chromosome?
Yes, and the gene for the tailmost structures is at the other end, with the genes for intermediate structures physically located in between on the chromosome.
Wow. That's incredibly organized.
It is. It shows this deep, conserved genetic logic underlying body plan specification across the animal kingdom. The basic toolkit and its organization are ancient.
This really reinforces the idea that evolution isn't constantly inventing brand new things from scratch.
Not usually at this fundamental level, no. It brings us to Francois Jacobs' famous metaphor of evolution as a tinkerer.
A tinkerer, not an engineer.
Exactly. An engineer might design something optimal from the ground up. A tinkerer, or bricoleur as Jacob put it, uses whatever parts are already available, modifying them, combining them in new ways to solve a problem. Evolution works with existing genetic pathways and networks, tweaking the regulation.
And because different lineages inherit the same basic toolkit, they can end up modifying it in similar ways to solve similar environmental challenges.
Which helps explain why we see evolutionary convergence so often, things like wings evolving independently in insects, birds, and bats. They're using underlying conserved developmental pathways, regulated differently, to achieve a similar functional outcome.
This idea of tinkering also fits well with modularity, doesn't it? The idea that organisms are built from semi-independent parts or modules.
Absolutely. Modularity is key. It means biological systems, genes, cells, tissues, organs, are often composed of these distinct subunits that can develop, function, and importantly, evolve somewhat independently. This is sometimes called mosaic evolution.
Can you give an example?
Sure. Think about the lower jaw in mammals, the dentary bone. It looks like one solid piece, right?
Yeah.
But developmental studies reveal it's actually built from about 6 different cellular modules. Modules for the incisors, the molars, the angle of the jaw, etc. And these modules are under partially independent genetic control.
Meaning you could change one part without messing up the others?
Potentially, yes. For instance, if you experimentally knock out a gene like MSX1, it primarily affects the development of the incisor and molar tooth modules.
Yeah.
But the rest of the jaw structure might develop relatively normally. This modularity gives selection specific targets to work on.
That makes sense. And there was another great example, the cave fish.
Ah, yes. The Mexican cavefish, Astyanax mexicanus, a fantastic natural experiment. You have surface-dwelling populations with normal eyes and pigmentation, and then multiple independently evolved cave populations that are blind and lack pigment.
A trade-off, basically. They lose sight but gain something else.
Exactly. They lose their eyes, which are useless in total darkness, but they have greatly enhanced senses of taste and vibration detection through their lateral line system.
And this involves regulating the same genes differently.
Precisely. A key gene here is sonic hedgehog, or shh. In eye development, shh expression needs to be carefully controlled. In the cavefish, shh expression is ramped up in a particular region during development.
Which does what?
It suppresses eye development. But that same network, involving shh, PAX6, and other genes, also influences the development of taste buds and the lateral line. So selection in the cave environment seems to have favored regulatory changes that downregulate the eye module while simultaneously upregulating the sensory modules useful in the dark. It's tinkering with the regulation across different developmental modules.
So evolution acts on these modules, tweaking their development via these conserved regulatory networks. Let's place this capability in deep time now. Animal life is ancient.
Very ancient. The major animal lineages started diverging way back in the Precambrian, maybe 750 million years ago. That's long before the famous Cambrian explosion that kicked off around 545 million years ago.
And before the Cambrian, there was that weird Ediacaran period.
Right, the Ediacaran Biota,
Yeah.
from about 635 to 542 million years ago. These were strange, often large, multicellular organisms. It's debated exactly what they were. Maybe early animals, maybe something else entirely. It's a sort of separate experiment in multicellularity, sometimes called the Vendozoa.
They had weird shapes, didn't they? Like quilted mattresses or fractals?
Some did, yes. Organisms like the rangeomorphs had these very repetitive, fractal-like modular constructions, very different from most animals we see later. Some might be stem taxa, early offshoots related to modern groups, while others might represent lineages that just went extinct.
It makes you think about all the possible ways to build a body. Paleontologists talk about morphospace, right?
Yes, the idea pioneered by David Robb. Morphospace is a conceptual, multidimensional space representing all theoretically possible forms or shapes an organism could take.
And where does actual life fall in this space?
That's the fascinating part. Known life, past and present, occupies only a surprisingly small, clustered portion of the potential morphospace. Vast regions of theoretical possibility seem to be empty.
Why? Constraints.
Likely a combination of factors. Developmental constraints, maybe some forms are just impossible to build with the available genetic toolkit. Functional constraints, maybe some forms just wouldn't work biologically. And historical contingency evolution works with what it inherits.
Okay, so then we hit the Cambrian, and suddenly, bang.
Yeah.
This explosion of diversity and new body plans appears relatively quickly in the fossil record. What drove that?
It was likely a perfect storm. Environmental changes were happening significantly, rising oxygen levels, changes in ocean chemistry. Ecological factors were kicking in, like the predator-prey arms race, driving selection for shells, skeletons, better senses, faster movement.
Escalation.
Right. But crucially, the biological potential was already there. The evolution of sophisticated gene regulatory networks, including those Hox genes we talked about, during the late Precambrian, provided the means for this diversification. The toolkit was assembled and ready.
So when the environmental and ecological conditions changed, life had the genetic capacity to rapidly generate new forms by tinkering with these existing regulatory systems.
Exactly. That ties it all together. The major engine driving the diversity of animal forms wasn't necessarily the invention of radically new genes, but rather changes in the regulation of ancient, conserved gene networks. Evolution as elegant tinkering.
It really highlights that the difference between a fly and a fish, or a fish and us, isn't so much about having fundamentally different parts lists.
Much less than you might think. It's about the sophisticated the timing, the location, the intensity of how that shared genetic toolkit is deployed during development.
That's a really powerful way to look at it. Thank you. That was a fantastic journey through some complex ideas.
My pleasure. It's a fascinating area.
And for you, our listener, here's something to think about, building on that idea of morphospace and conserved toolkits. If these regulatory mechanisms are so powerful and conserved, allowing for all this tinkering, why is so much of that potential morphospace empty? What really stops life from exploring those other forms? And perhaps, relatedly, how much of the story of evolution isn't just about change, but about the remarkable feat of simply staying the same, maintaining a successful form over vast stretches of time? Something to ponder. Join us next time for another deep dive.